Even simple motions make ripples across brain. Even simple motions make ripples across brain. You're listening to World Reading Club Technical News Reading, presented by Hakeem-Ali-Bocas-Alexander on Spreaker, social podcasting, presented for World Reading Club in association with UniquilibriuM. This edition's reading focus comes to us from neurosciencenews.com and is titled, Even Simple Motions Make Ripples Across Brain. It was published on January 27th, 2023. Summary. A simple motion, like a push of a button, can send ripples of activity across neurons spanning the entire brain, a new study reports. Source: University of Oregon. Even a simple movement like pushing a button sends ripples of activity throughout networks of neurons spanning across the brain, new University of Oregon research shows. The findings highlights just how much or just how complex the human brain is, challenging the simplified textbook picture of distinct brain areas dedicated to specific functions. It's really well known that the primary motor cortex controls movement output, said Alex Rockhill, a graduate student in the lab of human physiology professor Nikki Swan. But there's a lot more to movement than this one brain area. Rockhill is the first author of a new paper from the lab, published in December in the Journal of Neural Engineering. Swan and her team are studying brain networks in humans thanks to a collaboration with Oregon Health and Science University doctors and researchers. The OHSU team is using a technique called intracranial EEG to determine where seizures may be starting in patients with treatment-resistant epilepsy. They surgically implant an array of electrodes into partners' brains or into patients' brains to pinpoint precisely when and where a seizure is happening and potentially remove the affected brain area. Intracranial EEG also can provide valuable insight into other brain activity too. It's a gold standard technique, Swan said, but it's one researchers rarely have access to because implanting the electrodes is such an intensive process. Participants in Swan's study have agreed to let her team study their brains while they're already hooked up to electrodes for the seizure study. Swan and her colleagues gave study participants a simple movement-related task, pushing a button. They recorded the activity of thousands of neurons throughout the brain while participants were doing the task. Then, they tested whether they could train a computer to identify whether particular patterns of brain activity were captured while the participant was at rest or moving. In certain areas of the brain, the signals were obvious. Those were areas previously linked to movement, where most of the neurons are probably focused on that behavior. But the researchers also found brain signals predictive of movement throughout the brain, including areas that aren't specifically dedicated to it. In many parts of the brain, we can predict with higher than chance accuracy whether that data was from during movement or not during movement, Swan said. We found there's a spectrum of brain areas, from primary motor areas where you can decode that person is moving 100% of the time, to other areas that can be decoded 75% of the time, Rockhill added. There's a wonderful picture of a brain looking like a neural network with some circuitry involved. It's a digital image, and the caption reads, They recorded the activity of thousands of neurons throughout the brain while participants were doing the task. The image, which is also used here on Spreaker because it's in the public domain. Neuroscience News continues. In some of the areas that don't specialize in movement, some of the neurons might be firing, but they might be overwhelmed by neurons that are not movement-related, he said. Their findings complement a study published in 2019 in the journal Nature, in which other researchers showed similar far-reaching brain networks related to movement in mice. That paper showed that movement is everywhere in the brain, and our paper shows that's true in humans too, Swan said. The phenomenon probably isn't limited to movement either. Other systems like vision and touch are also probably extending through more of the brain than previously associated. Now the team is working on developing new tasks that involve different kinds of movement to see how those show up in the brain. And they plan to keep growing the collaboration with OHSU, involving more researchers in the project and gaining a deeper understanding of the brain's intricacies. There's a lot of opportunity now that we have this new collaboration, Swan said. We're really fortunate to be able to have the opportunity to collect such exciting data by working with the OHSU team and their incredible patients. About this neuroscience research news. Author Laurel Hamers. Source University of Oregon. And of course contact Laurel Hamers or the University of Oregon. The image referred to earlier, which is used here also on Spreaker, social podcasting, is in the public domain. The original research is under closed access and is from a paper titled Stereo EEG Recordings Extend Known Distributions of Canonical Movement-Related Oscillations by Alexander P. Rockhill and others, Journal of Neural Engineering. You've been listening to World Reading Club's Technical News Reading, presented for or by me, Hakeem-Ali-Bocas-Alexander, on Spreaker, social podcasting, presented for World Reading Club in association with UniquilibriuM. NeuroscienceNews.com returns or resumes with an abstract from the paper titled Stereo EEG Recordings Extend Known Distributions of Canonical Movement-Related Oscillations. And a sip of hot mess. Alright. Stereo EEG recordings extend known distributions of canonical movement-related oscillations. Objective. Previous electrophysiological research has characterized canonical oscillatory patterns associated with movement, mostly from recordings of primary sensorimotor cortex. Less work has attempted to decode movement based on electrophysiological recordings from a broader array of brain areas, such as those sampled by stereoelectroencephalography, SEEG, especially in humans. We aimed to identify and characterize different movement-related oscillations across a relatively broad sampling of brain areas in humans, and if they extended beyond brain areas previously associated with movement. Approach. We used a linear support vector machine to decode time-frequency spectrograms, time-locked to movement. And we validated our results with cluster permutation testing and common spatial pattern decoding. Main results. We were able to accurately classify SEEGs, which remember are stereoelectroencephalography, right? Stereoelectroencephalography. We were able to accurately classify SEEG spectrograms during a key press movement task versus the inter-trial interval. Specifically, we found these previously described patterns: beta, 13 to 30 Hz, desynchronization, beta synchronization, rebound, pre-movement alpha, 8 to 15 Hz, modulation, a post-movement broadband gamma, 60 to 90 Hz, increase, and an event-related potential. These oscillatory patterns were newly observed in a wide range of brain areas accessible with stereoelectroencephalography, EEG, that are not accessible with other electrophysiology recording methods. For example, the presence of beta desynchronization in the frontal lobe was more widespread than previously described, extending outside primary and secondary motor cortices. Significance. Our classification revealed prominent time frequency patterns, which were also observed in previous studies that used non-invasive electroencephalography and electrocorticography. Let's look at that again. Electrocorticography. Oh, that's right. But there were... Okay, let's start again. Significance. Our classification revealed prominent time frequency patterns, which were also observed in previous studies that used non-invasive electroencephalography and electrocorticography. But here, we identified these patterns in brain regions that had not yet been associated with movement. This provides new evidence for the anatomical extent of the system of putative motor networks that exhibit each of these oscillatory patterns. You've been listening to World Reading Club's Technical News Reading, presented by Hakeem-Ali-Bocas-Alexander on Spreaker. Podcast. Presented for World Reading Club in association with Uniquilibrium. This edition's reading focus comes to us once again from neurosciencenews.com and is titled, Even Simple Motions Make Ripples Across Brain. It was published on January 27th, 2023 on neurosciencenews.com. You can read this article and others like it for yourself by visiting neurosciencenews.com. That's neurosciencenews.com. That's neurosciencenews.com.